Guys, welcome to Smart Science Tutorial. Today we are going to discuss probiotics, the role of probiotics in promoting digestive health and overall well-being. So let's get started. Introduction. So probiotics are live microorganisms that, when ingested in adequate amounts, can confer health benefits to the host, particularly by improving or restoring the balance of the intestinal microbiota, the community of microorganisms in the gut. Probiotic are live microorganisms, so that upon digestion benefits the intestinal balance of the host. Probiotics and their benefits. So upon digestion, probiotic benefits the intestinal balance of the host. Probiotics help maintain or restore the balance of beneficial bacteria in the gut, which is crucial for digestion, immune function and overall health. When the balance of the gut microbiota is disrupted, it can lead to digested issues, infections, or other health problems. Probiotics can help by promoting the growth of the beneficial bacteria and suppressing harmful microorganisms. There are common probiotic genera. First one is Bifidobacteria and second one is Lactobacillus. So Bifidobacteria, a genus of bacteria that is frequently found in the intestines. Bifidobacteria are known for supporting gut health by aiding digestion, preventing harmful pathogens from colonizing the gut, and promoting immune health. And second one is Lactobacilli. Uh, Another major genus of probiotic bacteria, lactobacilli, are found in the small intestine and are known for their ability to ferment lactose, which is also called milk sugar, into lactic acid. This helps maintain an acidic environment in the gut, which can prevent the overgrowth of harmful bacteria. There are probiotic microorganisms. These are specific strains of microorganisms known for their probiotic properties. The Enterococcus species, these bacteria are found in the intestines and are involved in the fermentation of carbohydrates. Some strains have shown potential in helping with the gastrointestinal health. The second one is Escherichia coli. This is a specific strains. So, while some strains of E. coli are harmful, certain strains like E. coli Nesse 1917 have beneficial effects on the gut. These specific strains can help maintain a healthy intestinal environment and have been used to treat conditions like irritable bowel syndrome (IBS). The third one is yeast. So there are two examples of yeast. First one is Saccharomyces boulardii and second one is Saccharomyces cerevisiae. So these are yeast-based probiotics that help maintain intestinal health by preventing harmful pathogens from growing and promoting the balance of gut microbiota. Saccharomyces boulardii in particular has been shown to be effective in preventing and treating diarrhea, especially caused by antibiotics or infections. So expanding probiotic definition. Traditionally, probiotics have been associated with gut health, but recent research has expanded their potential applications. So, the first one is application beyond digestive tract. Probiotics are now being studied for use in areas beyond the gut, such as in vaginal health. Strains like Lactobacillus rhamnosus have been shown to help prevent bacterial vaginosis and yeast infections by promoting a healthy vaginal microbiome. And the second one is prophylactic actions of specific strains. Certain probiotic strains like Lactobacillus rhamnosus and Lactobacillus fermentum have uh, have shown potential in preventing infections and diseases. These strains have been researched for their ability to reduce the risk of gastrointestinal infections, urinary tract infections, and even some types of skin conditions. The last one is classification criteria. So, first one is beneficial effect established in vivo in living organisms. For a microorganism to be classified as a probiotic, its benefit must be demonstrated in living organisms, typically through clinical trials or animal studies. The microorganism must have positive effects on the health of the host, example, improving gut health, immune function, etc. And the second one is strain specific effects, not applicable to all probiotics. So, so there is example Lactobacillus rhamnosus GG has been shown to be particularly effective in preventing gastrointestinal infections, but other strains of Lactobacillus may not offer the same benefits. So, therefore, the health benefit of probiotics are probiotics are strain specific. So, meaning that it's important to use the right strain for the desired health outcome. So, the probiotics are live microorganisms that, when used properly, can benefit the health of the host, especially the intestinal microbiome. Their application is expanding beyond gut health to other areas like vaginal health, and their effects are strain specific, meaning not all probiotics will provide the same benefit. Types of probiotic products. First, fermented milks. Products like yogurt, kefir, and buttermilk, which naturally ferment using probiotic bacteria such as lactobacillus and bifidobacterium. Second one is dairy products, includes probiotic enhanced cheeses, ice creams, and butter, providing a convenient source of live bacteria. Fruit juice, non-dairy product probiotic options for individuals with lactose intolerance or dairy allergies, though stability of probiotics in acidic environments is a concern. Freeze-dried supplements, encapsulated probiotics in capsules or pills, often used as health supplements due to ease of storage and convenience. Fifth is powdered probiotics. Probiotics in powdered form used as additives in food or drinks, offering flexibility in consumption. So there are some challenges in probiotic products like survival of live bacteria. So ensuring that the sufficient live bacteria reach the consumer and survive digestion to exert health benefits. Second one is temperature sensitivity. So some probiotic strains, particularly anaerobic bacteria, are sensitive to temperature and oxygen, affecting their viability. Third one is refrigeration requirements. So many products like fermented milk need cold storage, increasing logistical and consumer challenges. Fourth one is strain efficacy after freeze drying. While freeze drying prolongs shelf life, some bacterial strains may lose efficacy during this process. There are some advantages of probiotic products like multiple strains. So, probiotics can be formulated with various strains to address specific health benefits such as gut health, immunity, or mental health. Second one is variety of food vehicles. So, probiotic can be incorporated into diverse products from dairy to plant-based options, catering to different dietary preferences. Third one is customized dietary management. Probiotic formulations can be tailored to meet individual health needs such as enhancing digestion or reducing lactose intolerance symptoms. Distribution considerations. Uh, Refrigeration requirements, maintaining the cold chain for refrigerated products. It's challenging and costly, especially in warm climates. Accessibility in developing countries. Limited refrigeration infrastructure in developing regions can restrict the availability and increase costs, making probiotic less accessible to many populations. So, while probiotic products offer diverse health benefits, their production, distribution, and consumption come with challenges that must be addressed are effective delivery. For effective delivery, survival of probiotic strains, probiotic survival requirements. First, which is like rigid stomach and intestinal conditions. Probiotic strains must endure extreme conditions in the stomach, low pH and intestines, bile salts, and digestive enzymes. Second one is survive transit to the large bowel. To provide health benefits, probiotics must remain viable until they reach the large intestine where they interact with the gut microbiota. There are some key considerations. First one is live probiotics are more effective. Only live bacteria can actively colonize the gut and confer health benefits. Dead or inactive cells lack the ability to replicate or interact metabolically with the host. In vivo survivability challenges. Probiotics face harsh conditions like gastric acid, bile salts, and pancreatic secretions, reducing their survival rate. Limitations of commercial testing. Most testing of probiotic strains occur in vitro lab conditions, which may not accurately replicate the actual conditions within the human body. Fourth one is strain variability. Different strains vary in their ability to survive. For example, strains of lactobacillus generally exhibit better survival rates compared to others. Human gut derived probiotics are superior. Strains like Lactobacillus rhamnosus, L. reuteri, and LGG originating from the human gut tend to perform better than strains derived from dairy or other sources. There are some essential factors for effective probiotics. Acid and bile resistance. Effective probiotics must resist acidic stomach conditions and bile salts in the intestines. Origin probiotics derived from the human gut are better adapted to survive and function in the digestive system. Strength specificity. The health benefits of probiotics depend on the specific strain as different strains have unique characteristics and functionalities. Live cells. Probiotics must be alive to exert beneficial effects. Dead or inactive cells are significantly less effective. In vivo persistence. Probiotics must persist in the gut environment long enough to interact with the host microbiota and provide benefits. So, in summary, the effectiveness of probiotics hinges on their ability to survive the harsh gastrointestinal environment with human gut derived acid resistant strains showing the greatest potential for health benefits. However, bridging the gap between in vitro testing and real life human applications is crucial for ensuring their efficacy. Probiotic persistence and safety. Dose effect. Studies have shown that a daily dose of 10 raised to 10 CFU, CFU is colony forming units, of probiotics such as Lactobacillus rhamnosus GG and Lactobacillus johnsoni lal is considered safe for consumption. This doses has not been associated with adverse effects in healthy individuals. Minimum effective dose for immune response. A daily intake of at least 10 raised to 10 CFU is required to stimulate an adequate immune response, ensuring the probiotics beneficial effects are realized, such as enhanced gut health and immune modulation. High dose tolerance, even at higher doses such as 3 into 10 raised to 12 CFU per day of VSL, a multi strain probiotic formulation containing 8 different probiotic strains. The probiotics were found to be well tolerated in patients with inflammatory bowel disease. This suggests that probiotics can be safely consumed at therapeutic doses significantly above the standard levels. Transfer of antimicrobial resistance. Caution with Enterococcus species. Probiotics containing Enterococcus species must be used carefully because they are known to naturally harbor antimicrobial resistance genes. This poses a risk as such genes could potentially be transferred to pathogenic bacteria in the gut. Risk to gut microbiota. The transfer of AMR genes from probiotics to other gut microbes could disrupt the balance of the gut microbiota and contribute to the global problem of antibiotic resistance. Probiotic strains should like probiotic strain selection. To mitigate risk, probiotic strains used in products should be susceptible to at least two commonly used antibiotics. This ensures that they can be controlled or eliminated using antibiotics if necessary, reducing the potential risk of AMR gene transfer. These points highlight the need for careful evaluation of the dose, persistence, and safety of probiotic strains used in health applications. Safety of probiotics. Probiotic safety positive aspect. First one is proven safety record. Probiotics, particularly established strains like Lactobacillus and Bifidobacterium, have a long history of safe use in both healthy individuals and certain patient groups. Low risk of like adverse events. The risk of serious side effects such as bacteria entering the bloodstream. Bacteremia means bacteria enters in the bloodstream, or intestinal overgrowth is extremely low in healthy individuals. Rare infection, third one. Probiotic associated infections are exceedingly rare even among those consuming high doses regularly. Low risk groups are Lactobacilli and Bifidobacterium. So, Lactobacilli strains like Lactobacillus acidophilus and Lactobacillus rhamnosus are well tolerated and widely used in dietary supplements and functional foods. Bifidobacteria, so these are considered safe and effective for promoting gut health, particularly in infants and older adults. There are some precautions. So, first one is emerging probiotics. The strains that are not extensively studied or newly developed probiotics should be evaluated for safety before being introduced into the widespread use. Second is severe medical conditions. Individuals with chronic illness such as severe pancreatitis should avoid unsupervised probiotic use as it may pose risk. Third one is immunocompromised individuals. People with weakened immune system, example due to chemotherapy or HIV, are at a higher risk of infection. So probiotics should be used cautiously under medical supervision. Exceptions: Enterococci and yeast. Probiotics like Enterococcus species or certain yeast, example Saccharomyces boulardii, carry a higher risk due to their potential for antimicrobial resistance, making them for unsuitable for some patients. Second one is consultation with healthcare professionals. Vulnerable groups, including the elderly, pregnant women, or those with serious medical conditions, should seek professional guidance before taking probiotics to ensure their safety and efficacy. So, by adhering to these safety guidelines and precautions, probiotics can be used effectively with minimal risk.